Hello, welcome, Bethany community. I'm very pleased to introduce today's guest lecture to kick off the exciting 2022 Bethany Connect Speaker Series. I have had the pleasure of knowing our speaker for over 25 years, since I first came to the National Institute on Aging as a postdoctoral fellow and then as a principal investigator. We are certainly in for a treat today as we hear from a renowned physician scientist who leads a research lab focused on understanding both the genetic and environmental risk factors that challenge healthy aging in our community. Our speaker is not only an accomplished scientist, she is an exceptional mentor. She is leading the charge to train the next generation of scientists and encourage them to set their sights high to excel in research, career ambition, and life. So, students, alumni, faculty, staff, and friends of Bethany, please tune in to learn about the secret sauce of healthy aging from an expert and an all-around good person on the planet. Dr. Michelle K. Evans, an internist and medical oncologist, received her undergraduate degree from Barnard College, Columbia University, and a medical degree from Rutgers, the State University of New Jersey, Robert Wood Johnson Medical School in Piscataway. She received her postgraduate training in internal medicine at Emory University School of Medicine and fellowship training in medical oncology within the medicine branch of the clinical oncology program at the National Cancer Institute. Dr. Evans serves as Deputy Scientific Director of the National Institute on Aging, as well as the NIA Training Director for the Intramural Research Program at NIA. Trained as a physician scientist, she conducts epidemiologic clinical research in health disparities and basic bench research on the biology of health disparities and minority aging. So we're so pleased to have you, Dr. Evans. We want to welcome you to Bethany College. And I will say that I just received your email with your slides. So if we haven't resolved that issue, then I can project those slides from here.
All right, so Bob, you're not seeing a full screen show. You're just seeing PowerPoint, the working version.
Yes. Yeah, we're still.
All right. So can you want to run the slides for me?
Let me try that. And then
let me stop the share and then see if you can share.
Okay. Okay.
All right. Good? I guess we're in business. Thank you, Bob.
Okay, so Michelle, you can just tell me to, like the old-fashioned days, next slide, please, or whatever you want.
Yeah, there we go. That's how we have to do it, but that's okay. I'm just so thrilled to be here. I would just really like to thank Dr. Joe Lane and Bethany alumnus and my dear friend and colleague of many years, Dr. Bob Brosh, for the wonderful invitation to present a seminar as part of your college convocation series. I remember when I was an undergraduate, I always enjoyed the convocations at Columbia because I always felt that I learned new things. So I hope that that is the case today for the attendees. And I think it's a particular honor today because this seminar occurs during your celebration of the Reverend Dr. Martin Luther King, who transformed American life overall and my life in particular. As many of you know, health disparities research is a very broad, it's a multifaceted field that includes clinical research, basic research, translational research, sociologic research, and And today I'd sort of like to share with you just how we sort of put our heads together to formulate an interdisciplinary approach that's targeted really to understanding the biologic transduction pathways through which these social determinants of health actually result in poor and disparate health outcomes. But I think we should start by sort of understanding, well, when I say the road to health equity, what is health equity? Next slide, Bob. So health equity is achieved when every person has the opportunity to attain his or her full health potential and has no disadvantage in trying to achieve that could be related to social position or other social circumstances. When health equity is impeded, next slide, please, health inequities and health disparities result. Health inequities really reflect the differences in things like length of life, the quality of life, rates of disease, aging, disability, death, and as well as even severity of the disease you wind up developing. Health disparities are those differences in health, and they can be related to the numerous social demographic factors that I've listed on the slide in blue. Next slide. So here I'm showing you as an example the life expectancy at birth statistics up to 2017. So if you're a Hispanic female, although they suffer significant health disparities, they actually have the highest life expectancy when we're looking at Hispanic origin, race, and sex. But it doesn't escape any of us that that very low bar in blue, non-Hispanic males, they have a much lower life expectancy at birth compared to Hispanic females, compared to Hispanic males, compared to everyone. Next slide, please. So you'll see there's a 14-year, almost 15-year age gap between African-American men and Latinx females, and a 7, almost 8-year gap for African-American men and Latinx men. So you may ask, next slide, please. Well, why am I interested in this? And Dr. Lane and Bob asked me to sort of weave in a little bit of biographical information. Well, I'm interested in it because I'm a daughter of the South Bronx. I grew up in the South Bronx, and next slide, please. And I'm interested because health inequities and health disparities are rampant in the South Bronx, and worse, rampant in the South Bronx when I was growing up there. And they're even worse post-COVID. So that's why I sort of developed this interest and decided to devote my career to this. Next slide, please. So I grew up in the New York City Housing Authority public housing, the projects, in America's poorest congressional district. But from America's poorest congressional district, next slide, please, I found myself doing my undergraduate work at Columbia University. And at each of my stops, I was able to find mentors who I think recognized my potential and supported my dream of going into medicine. Although at that time, I don't know that I was thinking about biomedical research, but I wanted to have a career in medicine. And for the students here, I will have a picture of the important mentors at each of these steps, because at each of your educational locations, your educational institutions, you will find these people. And you have to recognize that they're out there. So never feel that, well, there's nobody here who wants to buy into my dream. They're there. You just have to find them. Next slide, please. I then went to Rutgers for medical school, and I had a number of mentors. I think maybe most significantly, Dr. Jack Gardner. Next slide, please. And then for internship in residency in internal medicine, I went to Emory, where I had a panoply of mentors who I think recognized my talent and at this time said, you should think about going into research. So this was the first place I really thought about, well, maybe I could think about a career that involved research. Next slide, please. And then finally, I sort of stumbled my way to the National Institutes of Health, where I just was really helped and guided and developed by these two men who sort of bought into the idea that NIH needed to recruit and support a diverse cadre of biomedical researchers. And I will point this out to you for minority students who may be in the audience. You will find many people who are not minorities who are willing to reach out their hand and help you. And so you have to recognize that. There are talented and extremely competent people who will buy into your dream, but you have to reach out. And I think that goes for all students, but I think particularly minority students feel sometimes that, well, I only can have a minority who's going to understand me. Well, yeah, there's some things that, yeah, I'm only going to understand because I lived that life. So I understand where you're coming from. But there are other people who want to learn from your experience and make it part of their mission in life. So it's a two-way street. So take advantage of that. Next slide, please. So I was developing as a basic scientist who did clinical oncology research. I became a medical oncologist in the National Cancer Institute and sort of was performing pro forma, doing the biochemistry of DNA repair as it related to cancer and cancer-prone illnesses. Next slide, please. I developed my own laboratory and again started doing different types of research related to cancer with a particular interest in breast cancer cell lines. And by this time I had moved from the main campus in Bethesda to the National Institute on Aging, which was in Baltimore. Next slide, please. However, next slide, next slide, next slide. So what I was driving past every day to get to work in Baltimore looked just like where I had grown up in the South Bronx. So it was one of these Yogi Berra deja vu all over again moments, sort of filled with these broken promises. You know, a daughter of the South Bronx starts to work in Baltimore and is reliving her childhood driving back and forth to work. And because I had lived and I also worked in the South Bronx, before I came to NIH, I took off 2 years and I worked as a primary care internist in the South Bronx, as well as in some underprivileged places in Brooklyn. And I just had an indelible recollection of these false promises, these broken promises. So that's when I decided to sort of take my experience in health disparities, my personal experience in health disparities and health inequities, and sort of reconnoiter my career. So next, I guess, pearl to pass on is you may start in one direction, but sometimes you sort of have a course correction, and that's okay. That's okay. Sometimes you have to step out on faith to do what you think maybe you promised to do. And you have the skill set and the environment to accomplish that goal. Next slide, please. So I went and started to look at what are the health trends in Baltimore, and I knew they were bad. Next slide, please. So in the United States, the overall life expectancy at birth is about 79 years old. Next slide, please. But in Baltimore at 73.6 years, next slide, please. It's comparable to people who are living in Azerbaijan. Next slide, please. And if you live in a red neighborhood in Baltimore, where the Blue Star is, It's comparable to those people who are living in Yemen. And remember, Yemen is in the middle of a civil war. Yeah, this is not good. In the in the green neighborhoods in Baltimore, Roland Park, next slide, please. Life expectancy even exceeds what we find in the US overall. So I felt committed to at least study this as a problem. Next slide, please. And so with my colleague, Alan Zonderman, as we were thinking about this problem, we designed the Healthy Aging in Neighborhoods of Diversity Across the Lifespan study known as HANDLES. So HANDLES is an interdisciplinary community-based longitudinal epidemiologic study that examines the interaction of race and socioeconomic status from the development of age-associated health disparities in a socioeconomically diverse cohort of African Americans and whites who reside in the city of Baltimore. We were trying to disentangle race and SES to understand how they influence, in what context they influenced health and differences in health, in rates of aging and the development of age-associated disease, and also to try to tease apart important biologic mechanisms or biomarkers that might be important. Next slide, please. So HANDLES is an area probability sample of the entire city of Baltimore based on the 2000 US Census. Our survey statisticians, using the data from the US Census in 2000, identified 13 census segments that would yield a representative distribution of the city of Baltimore and have sufficient individuals to fill the sampling frame, which was whites and African Americans, men and women between the ages of 30 to 64, who were above or below the 125% poverty line as stated in the federal poverty line set by the Department of HHS in 2004. So the goal was to recruit somewhere near 4,000 people, follow them for greater than 30 years as a fixed cohort, meaning we wouldn't be having new participants join the study, so that we could sort of examine trends in health, drivers of health disparities. Next slide, please. So to do this, to get around the city, we developed mobile medical research vehicles, 3 of them as a matter of fact, that serve as community-based research platforms that go to the different locations in the city where we have enrolled participants. Next slide, please. We were fairly successful in that we were able to recruit over 3,700 individuals. Next slide, please. And we have been following them since 2004. Of course, with COVID, we've had an interruption in parts of the study, but we're hopeful to resume and begin our next longitudinal wave shortly. Next slide, please. So we consider the social determinants of health as agents and toxic agents, actually, that work through biologic transducers that result in the accelerated aging phenotype, which is, we feel, the phenotype of health disparities. So you develop age-associated disease quicker, related to adversity over the lifespan, and that leads to more morbidity, disability, and ultimately mortality. So there are lots of different biologic pathways that may be important, and we sort of examine these in our laboratory. And they include, next slide, please, genetics, next slide, please, inflammatory pathways, next slide, please, oxidative DNA damage and repair, next slide, please, epigenomics, next slide, please, as well as examining gene expression. Next slide, please, and immune factors that may be important. Next slide, please. So we also consider race And race is important in health disparities research because it is not a biologic construct, it is a social construct. It's a social category that's been forged historically. It shapes human experiences because it fuels social exclusion and confers social privilege. Societies use race to establish and justify systems that convey power, privilege, disenfranchisement, and sometimes even oppression. And it's meaningful for health because it's a risk factor for poor health outcomes in the context of social status, an individual's ability to access resources, particularly. Next slide, please. Next slide, please. It won't go.
It's frozen right now. Oh, there we go.
Okay, so what are the outcomes when the social determinants of health get under the skin? Next slide, please. So if we look at things like income, next slide, please, or income inequality or poverty. Next slide, please. Next slide. So here you can take a look at just some data I thought would be illustrative of, the differences we're talking about. We're looking at real median household income between 1967 and 2020, and this is Census Bureau data. So we can look at the fact that it's more than a $45,000 difference in real median income between the highest income earners and the lowest. And African Americans and Hispanics since 1967 have been the lowest in terms of real median household income. Next slide. In Baltimore, the poverty rate is about 23%. Next slide, please. However, among African Americans, it's approximately 26%. So these are not 1 year of data, not 2 years of data. This is going back to 1967. So when we're talking about income inequality, these are long-standing issues associated with health. Next slide, please. So we decided to ask the obvious question to us was, all right, well, how does poverty relate to chronic disease? And we looked at diseases that are associated with a higher incidence, morbidity, and mortality among African Americans. One of them is chronic kidney disease. Next slide. So we looked at, next slide, we looked at chronic kidney disease prevalence in the Handles cohort, and then really didn't find that kidney disease, chronic kidney disease was more common in African Americans or whites. In this relatively young cohort, it's approximately equal. Next slide. Next slide. However, when we looked at the degree of illness or the severity of illness, African Americans were 5 times more likely to have advanced CKD. Next slide, please. And when we looked at chronic kidney disease prevalence in the context of SES, we found that there was a 27% greater prevalence of chronic kidney disease among the people in the cohort, Black and white, who were low SES. So again, showing that link between poverty and a chronic illness. Next slide, please. However, when we looked at it in the context of race, low SES was associated with a greater prevalence of chronic kidney disease in African Americans, but not whites. Next slide, please. Suggesting that poverty is a particularly virulent risk factor for chronic kidney disease in African Americans, but not for whites. Next slide. So disparities in brain health are increasing in the US population. When we look at individuals who are over 65 years old and older, African Americans have the highest prevalence of Alzheimer's disease and related dementia, followed by Hispanics, about 13.8%, 12.2%, somewhere in there, higher than whites and Asians. So we decided to understand or to examine whether or not there was a relationship between race, socioeconomic status, to global brain outcomes. So we put people in MRI machines to measure the volume of different parts of their brain. We also measured white matter lesion volume, which are lesions in the brain that are predictors of future poor brain health outcomes. And what we found was that there was a significant interaction between race and SES. Next slide, please. Among whites, when we looked at total brain volume, next. When we looked at white matter volume, next, and gray matter volume, white participants with higher SES had greater brain volumes than all other groups, although they were within the normal range. Next slide, please. African Americans with low SES exhibited significantly greater white matter lesion volumes than white participants with low SES. Next slide. And you can see that at the bottom there in the red boxes. So this again shows that poverty, low SES, is associated with not only brain volumes, but also lesions that develop in the brain with age that are associated with dementia and other poor cerebrovascular outcomes. Next slide, please. So the findings could reflect environmental or interpersonal stressors. You know, does stress play a role? So poverty is a life adversity that causes stress. And so maybe people who are African American or people who are poor have greater stress to deal with. So we wanted to understand, is there a possible way that other parts of the brain that we had not looked at initially in the MRIs, could they also be affected by race and socioeconomic status? And so we decided to look at the cortical limbic system, because the translation of stress and emotion to disease involves this bidirectional communication between different parts of the brain, particularly several cortical and subcortical brain regions, like the medial prefrontal cortex, the orbital prefrontal cortex, as well as the amygdala and the hippocampus. So this allowed us to look at the cortical limbic system, which processes a broad range of behavioral and cognitive function, trying to examine these relationships using regional brain volumes that were derived with a T1 weighted MP range image. Next slide, please. And what we found was, in fact, that socioeconomic status and race interactions were present in a number of the brain regions that we studied, including the right medial prefrontal cortex, the left medial prefrontal cortex, the left orbital prefrontal, as well as the left anterior cingulate cortex. High SES whites had greater volumes than all the other groups. And so this is another example where it's not just in gray matter and white matter, but it's in specific parts of the brain that both race and socioeconomic status influence brain volume. Next slide, please. So higher levels of chronic stress, which we know have been associated with low SES low SES individuals and African Americans, may contribute to race-related disparities and SES-related disparities in brain health outcomes. Next slide, please. So these findings about the relations between brain volume and poverty led us to go ahead and examine another group of Handel's participants who were at particular risk for decreases in cognitive function, those with diabetes. And you all know in the United States and worldwide, the prevalence of diabetes has increased dramatically. So we wanted to look at the cohort in the context of those with and without diabetes and evaluate 11 cognitive tests. Next slide, please. And what we found was that there were significant associations between the presence of diabetes and cognitive function. However, we only found it in African Americans who were living below poverty. And the associations were all negative. So diabetes was associated with poorer verbal memory, working memory, and attention among African Americans living in poverty. And what is important about this study, and for us to think about in the nation overall, is that poverty is influencing cognitive function in diabetics. And as you know, type 2 diabetes used to only be seen in older individuals. We now have young children and teenagers, because of obesity, being diagnosed with type 2 diabetes. So this is not just going to be a problem for African Americans. This may well be setting the stage for increases in cognitive problems in mid and later life that we, I think, epidemiologically certainly would not have predicted. Next slide, please. So we know poverty influenced chronic kidney disease, white matter lesion volume, total brain volume, cortical limbic structure, cognition in people with diabetes. Well, what's the most important outcome we're all interested in? It's how long are you going to live? You know, what's your risk of dying? When we examined poverty in the cohort, African American men who were living below the 125% poverty line had a markedly increased risk of mortality. So there was a three-way interaction between sex, race, and poverty status. African American men below poverty were 2.7 times more likely to die than those living above poverty. So that's a within race difference. It's a social factor that's driving that mortality. When we looked at white men, next slide, please. White men who were below poverty had about the exact same risk as those who were above poverty. And when we looked at, next slide, please. When we looked at African American women and white women who were below poverty, they had an increased mortality risk, but the risk was about the same. Next slide. So again, this is, I think, evidence that number one, poverty is more virulent for Blacks than whites, and we need to figure out why. African American men should be a target to be considered when we're trying to discern poverty-related transduction factors. Next slide, please. So we decided we wanted to understand, well, how do social factors somehow actually change biology? You know, so social genomics allows us to study the mechanisms through which social environments and exposures modulate genomic activity. Social environments influence gene expression via physical environmental exposures that can trigger a number of different responses, such as engaging cellular receptor systems and activating intracellular signal transduction pathways. Next slide, please. And so we were interested in sort of understanding transcription and gene expression. So we focused on RNA because there is a literature that has defined the conserved transcriptional response to adversity, where they've observed a pattern of gene expression that is predicted to occur because of exposure to adversity. Next slide, please. So we looked at RNA. Next slide, please. And as you know, there are many different types of RNA. Many of you in class, I'm sure, have studied mRNAs and maybe tRNAs. We looked at long non-coding RNAs because they regulate gene expression. Next slide, please. Now, there's a lot that's not known about the functional roles of long non-coding RNA, but we decided to look at them because we know that they're deregulated in some neurodegenerative diseases, including Parkinson's disease, as well as Alzheimer's disease. They're involved also in innate immunity, as well as cancer initiation and progression. But what's important is that they regulate gene expression, so they could change a biologic pathway. So in men in the handles cohort, we looked at white men and Black men who were above and below the poverty line, who were younger and older. Next slide, please. And we did find substantial changes in these long non-coding RNAs abundance with age, and there was a difference by race. As you can see in the top panel on the left, white males had many more link RNAs that were substantially changed with age than African Americans. Over 1,900 in white males, only 26 in African Americans. Next slide, please. We also saw that poverty significantly influenced link RNA expression. Again, mostly in white males. 1807 link RNAs were changed in expression compared to only 245 in African American men. Next slide, please. We also looked at messenger RNA abundance, and again, found the same thing. There was an alteration with poverty in both white men and African American men, but certainly more so in the white men in the cohort. Next slide, please. We went ahead and actually validated this expression by real-time PCR and found that many of the link RNAs that were associated with poverty played a role in tumorigenesis, in tumor suppression, and regulation of p53 target expression. So these are very novel findings, but we also did find some genes that were associated with that conserved transcriptional response to adversity that's been described in both men and in mice. Next slide, please. So, I mean, the bottom line of this study is that age and poverty as a marker of social adversity increased the expression of an mRNA, which is one of the things we wanted to dig down on. How does poverty cause disease? Well, if you live in poor housing, you don't have good food to eat, but it also changes some of the biology. So that's a direct environmental influence on things that are related to disease. Next slide, please. Next slide. So we looked at poverty. Next slide. So what about discrimination? Next slide. Next slide. So perceived discrimination, next slide, is important because it too is a psychological stressor and is associated with high blood pressure, obesity, and a number of other negative cardiovascular outcomes. Next slide, please. We measure perceived discrimination almost as a vital sign in the questionnaires that we have participants respond to. We ask them overall, how much harder has your life been because of discrimination? Overall, how much has discrimination interfered with your life? We ask specifically about racial discrimination and whether respondents have experienced discrimination at work, at school, getting a job, getting medical care from the police or in the courts. Next slide, please. So we first took the idea into the laboratory to see, could we come up with a biomarker that somehow linked discrimination to something that we could physically measure in the blood? And so we decided to look at red blood cell oxidative stress because it's been reported in many age-associated diseases and even with aging overall. And since we're trying to look at an aging related phenotype, accelerated aging phenotype, we thought this would be a reasonable marker. And we looked at red blood cell oxidative stress, which really is a measure of how hemoglobin undergoes autooxidation. And this is part of the normal redox reactions that happen with hemoglobin. So this autooxidation results in heme degradation products, which are fluorescent and therefore can be relatively easily measured. And we selected this particular marker, fluorescent heme degradation products, because they're sensitive to blood flow changes and tissue oxygenation that can be triggered by psychological stress through your sympathetic nervous system. So we looked at this cross-sectionally in the Handles cohort. Next slide, please. And found that discrimination was not associated with RBC stress in whites, but only in African Americans. Next slide, please. And we also looked to see, well, how was this influenced by other things that could contribute to the development of oxidative stress, such as age, smoking, obesity, or many inflammatory markers, and the relationship remained statistically significant. So we were able to see that those who reported high levels of discrimination, if they were Black, they had higher levels of red blood cell oxidative stress as measured by heme degradation products. Next slide, please. So we looked at renal function in poverty. So we also wanted to know, well, does discrimination influence renal function? Next slide, please. So we did a longitudinal evaluation of Handles participants looking at the renal function that we can get just from a simple blood test. Next slide. Next slide. So we also in this study looked at gender discrimination. So perceived gender discrimination was associated with lower renal function to start with in all participants. Next slide. Among white women, those who reported having a high experience of discrimination, that was also associated with lower baseline eGFR. Next slide, please. When we looked at African American women, both racial and gender discrimination were associated with a decline in renal function over the longitudinal time point. So this sort of shows you that discrimination is bad for everybody. And even gender discrimination can have an effect on a biologic function that is important to maintain good health. Next slide, please. So because we know about the disproportionate burden of poor health outcomes in African Americans, we wanted to understand the role of discriminatory stress in brain health. So here we looked only at African Americans who had had MRIs. And again, we looked at discrimination burden and age and white matter volume, white matter lesion volume. So if you look at the slice of the brain on the MRI, you'll see these little white cotton tufts. Those are the white matter lesion volume, white matter lesions that are focal areas where you have demyelinated cells. It's a non-specific injury, but it's a degenerative injury in white matter and is associated with cerebral small vessel disease and certainly increases the risk for developing dementia. Next slide, please. And so these results were a little confounding. Only part of our hypothesis here was true because there was a nonlinear association of discrimination to white matter lesion as a function of age. Kind of in keeping with our hypothesis, as you got older, if we look at the dotted lines, the 60-year-olds and over, as racial discrimination and lifetime discrimination burden increased, so did white matter lesion volume. However, if we look at the dashed lines for the 40-year-olds, mean age 40-year-olds, younger African Americans who did report greater exposure to discrimination, they had less brain pathology on MRI. So what does this mean? The effects of discrimination on brain health may be related to which age cohort you're in. There may be a threshold that has to be reached before discrimination results in biological change to the brain. We're doing follow-up studies to try to investigate this, but we do think the data certainly strongly suggests that discrimination and discrimination burden may play a role in the disproportionate burden of poor brain health in African Americans. Next slide, please. And we also looked at a subclinical marker of cardiovascular health, because as you know, African Americans have more hypertension, more cardiovascular disease. And so we looked at a subclinical marker of coronary risk in the context of discrimination and depression, and used the marker of carotid intimal medial thickness. And you'll see between the two arrows, we measured that very thin medial layer in the arterial wall of the carotid using high resolution B mode ultrasonography. Next slide, please. And what we found was that in African Americans who reported a higher lifetime burden of discrimination and also had greater depressive symptoms, they had higher levels of carotid intimal medial thickness, which is a known risk factor for hypertension, cardiovascular disease, as well as cerebrovascular disease. And we did not find a significant association among the white participants in the study. Next slide, please. And so when we're talking about aging biomarkers, and this is something that Bob and I hear all the time because we're in the Aging Institute, telomeres. Telomeres are those ends of the chromosome. So look at the little red caps on the purple chromosomes. Next slide, please. And so telomeres are short nucleotide sequences that are at the end of all linear chromosomes, and they're the protectors of the genome, preventing genomic instability. Genomic instability is a bad thing because it's associated with cancer. It also goes along with aging. Next slide, please. So long telomeres are associated with health and longevity, short telomeres with age-related disease and reduced longevity. And there is evidence that environmental factors as well as biological factors contribute to telomere shortening. So since stress and mental stress in particular has been suggested to be an important driver of telomere shortening, next slide, we looked in the Handel's cohort and we measured peripheral blood mononuclear cell telomere length by quantitative polymerase chain reaction. Next slide. Next slide. And what we found was that African American women overall had shorter telomeres than men. African American women who had greater lifetime burden of discrimination, racial or gender discrimination, also had shorter telomeres. But also men and women who were African American who were higher SES and reported a greater lifetime burden of discrimination also had shorter telomeres, as well as younger adults who reported exposure to multiple sources of discrimination. In whites, among white women, where there was no effect between racial or gender discrimination and telomere length. However, in men, we had a somewhat perplexing response. Among older white men who reported racial discrimination, they actually had longer telomeres, whereas younger white men had shorter telomeres. So this again is a mixed picture, and and I talk about this to let everybody who's thinking about a career in research, you don't always get what you expect, and you have to interpret the data to the best of your ability, but also do supplementary studies. So we don't know why we got these results in white men, although we actually, I think now, are sort of thinking about this in a somewhat different way. So we think it's possible that discrimination may contribute to telomere shortening that could lead to the accelerated aging phenotype associated with health disparities, particularly in African Americans, because there is data that has shown that telomere shortening can occur in caregivers who are taking care of disabled or Alzheimer's patients. So we know that stress is a factor. We're saying this is another factor that could perhaps promote the accelerated aging phenotype. Next slide, please. So, in this talk, we've looked at poverty, we've looked at discrimination, and, we've been able to find their influence on a number of different outcomes, from chronic kidney disease to brain volume to differential gene expression, white matter lesion volume, and oxidative stress. In our lab, next slide, please. We have looked at this problem from a number of different aspects. And I think that's the difficult part of health disparities research, is you have to put on a different pair of glasses to kind of come up with the different ways to examine a very complicated social and biologic problem. Next slide, please. Health disparities research is sort of a somewhat new field, in that we are all still trying to document the existence of health disparities in specific populations. So there's still primary work that's going on. We're trying to explain the reasons for health disparities, and then we hope to be able to provide solutions, but I can't say we're at the third generation of studies. But that's the goal, is to get there. Next slide, please. So, to be on the road to health equity, I think we have to understand that social factors cause disease, and that can come through biologic pathways. I think we all have the responsibility to be active in making sure we understand what the drivers are of health disparities, one of which is adequate, affordable health care for all. But I think we also have to recognize and combat structural and institutional racism wherever we find it, because it's bad for everybody. It has a negative effect on health. Next slide, please. And so a lot of the work that was done today, some was done by Deirdre Cruz, who we've worked with for many, many years. Danielle Beatty-Moody in the discrimination work, Doug DeLuzin and his team have contributed to some of the work in gene expression that I didn't have time to talk about today. Next slide, please. Of course, my co-principal investigator, we have one brain and we just slice it up differently every day. This is, I think, a very good example of true team science, people from two different backgrounds coming together to pursue a complicated interdisciplinary problem. Next slide, please. And then to everybody who makes Handel's work, Nikki Norenhooten, who's my staff scientist, has been critically important in a lot of the work. I talked about her linked RNA work, our physician, Ngozi Ejiogu, who is the one who sees all of the participants, as well as just our very diverse, hardworking group who keep a very difficult study afloat, and I think doing good for our participants who are our partners in trying to uncover the drivers of health disparities. Next slide, please. And that's it. Thank you very much for your attention.
Oh, thank you, Dr. Evans. Thank you so much. Your talk was just wonderful for our Bethany College community, but also thank you for modeling such interdisciplinary work for everyone beyond our community. I was really inspired by that. I wanted to open our question session by asking you one myself. Just where would you begin the low-hanging fruit? I mean, given the oxidative stress work and the telomere length and all these different possible markers that you could use, but where would you like to see routine tests, something changing that doctors would start to do, maybe even asking surveys about racial and gender lifetime burdens? I mean, where would you begin that would be affordable, that we could start there?
Yeah, I mean, I think we've talked about this in some of the work I didn't talk about today. Something as simple as looking at on your complete blood count, there is one of the categories of results is called the red cell distribution width, or RDW. And physicians don't do anything with that. But our study, as well as many other studies, have shown that it's associated with mortality. So why are physicians not using that? Every time they get a complete blood count, can you look at that number and say, well, gee, maybe I need to investigate this person a little bit more. They came in for A, B, and C, but this result could suggest that they're at higher risk for mortality. Maybe I should examine why. You don't have to spend another dime. You just have to use what you're getting. Other things, screening for discrimination, screening for depressive symptoms, these are easy to add on things. And in some practices, physicians are beginning to do this, but as a physician myself, it's like we're in this time crunch where you're supposed to see X number of patients in a very finite period of time to keep things moving. It really gets very difficult. I think we have to almost change the model of care. And there are some practices that are doing this, but also using nurse practitioners as really physician extenders is also critically important, because it's not, I think we have to think about medicine in a slightly different way, not so much top down, but more planar in terms of physician extenders and what they're able to do and what information they need to feed back to physicians.
Absolutely. Thank you so much. Let me read off of our chat. What are some questions others have submitted here? From Walt Hunt, we have you studied a role of obesity, poor diet, lack of exercise, and diabetes in health disparities?
Yes, yes, and yes. And I guess some of the interesting findings, certainly relate to how people eat. And our healthy eating index, which is sort of a measure of how many healthy foods you're eating, is quite low in handles. And that certainly contributes to obesity, which then contributes to some of these other diseases. So it's a little bit of a chicken and egg thing. What came first? Well, did the poverty come first? And you're living in a poor neighborhood, you can't get healthy food, or the cheapest food that is available to you is very calorie dense. And we also have people who are food insecure. So you would think, well, food insecurity means they don't have enough food to eat, they don't have enough money, so they're going to be very thin. But no, because they're going to that dollar menu at McDonald's. So, because they can get 3 things, so that's $3. That's a cheap meal, but those things are incredibly calorie dense. And again, these are the things that, drive health disparities. That's why understanding the importance of the social determinants of health in these disparate health outcomes is just critically important. And we try as much as possible to model healthy eating through our newsletter, through the meals we feed participants when they come to participate in the study. We send out in our newsletter healthy recipes. We have done everything to try to model better eating. But that's also something that many didn't even learn in school. And if you look at the most recent data that's come from Case and Deaton, the effect of a college education on longevity is dramatic in both African Americans and whites. And so education remains an incredibly important driver of better health. And that's another thing I think we have to consider, as we're thinking about, well, how do we get the largest number of people to community college, at least, to have some college, because that has a direct impact on health.
Thank you. All right. Next question was, what are the criteria to define discrimination among these subjects?
Well, we use validated questionnaires. And remember, it's the participant or the individual's perception is how is it that they're feeling. And what one may describe as discriminatory may not be discriminatory to another. So so that's why we only really use the validated questionnaires based on their reports. So because again, just like when you talk about gender discrimination or sexual harassment, this is who's experiencing that. So we allow our participants to give voice to what it is they feel has impacted their life. And that's the basis of, discrimination research.
Yes. All right, I have a question from Dr. Jolene, our provost. He said, Dr. Evans, thank you so very much for this amazing talk. We are very grateful for your sharing this with our Bethany communities. I wonder if you might be willing to discuss a little bit about any of the ways that public policy decision makers or agencies are accessing and using this research in making their public health policy decisions. Thank you very much.
Well, every chance we get, we're sharing our data. At NIH, health disparities research, has now become a highlighted area of research in terms of trying to generate larger program portfolios of this research. So, and some of it is based on our findings as well as the findings of others. So I do think it's beginning to have an influence. It's been a steep climb because many thought, well, this is really a social agenda or a political agenda. No, these are facts. And I think that's one reason why I wanted to do some laboratory-based research to really show the impact on health that people don't have control over. There are some things people can control, but many things they can't. What's going on in their neighborhood, many of them can't control. And so these things have negative impacts on their ability to achieve health equity. If where you live, you have to take 3 buses to get to the doctor, okay, well, how is that your fault? But that's going to have a negative impact on you being able to access treatment. And that's going to be something that impacts your ability to get to health equity. And so these are sort of the many different pathways we've in our lab taken, but there's a large community of investigators who have tried to come at it from different places. And I do think that the federal agencies are sitting up and taking notice and heightening their commitment in terms of dollars, in terms of resources to address these issues.
Oh, I hope so. I hope so. Here is Martin Jones asking, thanks for the wonderful talk. Going off your response to Walt Hunt, it seems that great interdependence between race and SES makes a statement of directionality very challenging. From an epidemiological and biological perspective, what have been the challenges you have experienced when trying to separate or disentangle these two parameters?
Yeah, it's very difficult because some people do not give you their true income, or they'll give you ranges of their income. And so, yeah, trying to do that, I think, is difficult with white participants as well as with Black participants. And I think some of our data that certainly suggests that poverty has a greater influence on health in certain circumstances in African Americans compared to whites, certainly, suggests that there are some differences. We can sort of disentangle it because in having CKD in our cohort, well, there was a racial difference in who had it or who didn't. In severity, yes. But when we looked at it, it was really the people who were poor and Black. It was an additive effect. So, but doing further disentanglement and trying to study income, trying to study occupation, these are very difficult things that we've had difficulty, I think, understanding, because if we look at household income, that's one thing, but we have not even been able to put our hands around the issue of wealth, because your income, your household income is really only one facet of your socioeconomic status. There are people, who have very little income. I think, does Bill Gates take $1 a year in income? But his wealth is tremendous. So trying to measure wealth, measure home ownership, those things that would really, I think, refine our understanding of someone's economic position. We're not doing a good job at that. I'll raise my hand and say we've done the best we can, but it's not perfect. And I think other investigators who work more specifically in this area would tell you how difficult it is to understand wealth. A question like who has credit and who doesn't. Having credit allows you to access more resources. But if you don't have credit, then you're in a different economic strata. So even trying to examine that is complicated.
Yes. Ethan Schaefer has asked, do these studies include undocumented immigrant populations in Baltimore City, especially in high density locations such as Highlandtown? And do you think this document status influences health statistics apart from race?
Well, it's interesting you should mention Highlandtown because we are located. That's where the NIA and the National Institute of Drug Abuse are located on the Hopkins Bayview campus. So we do have participants in Highlandtown. However, we had people self-identify. So did you identify as white or African-American? Because we were just doing a biracial study. In 2000, there were not enough Hispanics in Baltimore, documented or undocumented, to really be able to do that group in the study, because that was one of the things we wanted to do, was to look at Hispanics, African Americans, and whites. But based on the 2000 census, there just weren't enough Hispanics for us to get a representative distribution. So we did not include them. Now, as you mentioned, there are many more Hispanics in Baltimore now. So if we were doing the study now, we probably would have approached it differently.
Dr. Brosh has asked, do you plan to examine the relationships of COVID-19 with society factors, race, et cetera, as they impact accelerated aging?
Yes, we were out in the field doing COVID testing. So we do have a small COVID cohort that we've been following since the pandemic began. And so now the issue is now trying to sort of get back out there. You know, we've been out there doing small portions of the study. With Omicron, we've sort of stepped back, trying to sort of let that wave go down before we go back. But yes, we will be collecting information that's particularly relevant to COVID, because if you look at the latest statistics in terms of life expectancy, African Americans have lost more than 3 years of life expectancy. I think Hispanics have lost 2.2 years of life expectancy related to COVID. So, you know, so even in terms of life expectancy, it's had a major effect. And we think it will also have effects in some of the other areas which are health disparities related conditions.
We've got lots of questions for you. I don't know if we're going to have time for all, but let's read this one from Lisa Calamaro. Circling back to the portion on diabetes and maybe Parkinson's, does childhood poverty or stress versus sustained poverty make a difference? If someone comes from poverty but raises up to affluence, do they carry that outcome with them?
That's an excellent question. And I think we don't know the answer. What we have added to, and this is maybe 5 years ago, we started collecting data on adverse childhood experiences because we wanted to be able to ask those questions and then look at it in the context longitudinally of what happens to people. And so we do think there is an effect, but what is it? And are there ways to be resilient against early life negative exposures? We don't know, but that's a very, very good question.
I had one last one, and folks, keep adding them if you're good to stay, and if you can stay, Dr. Evans, we'll keep going. But I had a question for you about advice for our undergraduates, because I know they're coming from a variety of different majors, and the teamwork that you just described for us between people who had different backgrounds and trainings and probably different degrees. I thought maybe you could help describe a bit of those different training paths that the students might want to take towards an MPH or an MD or a PhD if they're coming, even like data management. We have computer science majors, math majors that might want to come in and be part of these teams. And if you could describe how you'd get there, like get where you are.
Well, all of the above. I mean, I'm a medical oncologist with an MD. I didn't have laboratory training until I came to NIH. My co-PI, Alan Zonderman, is a neuropsychologist with a PhD. But we have people from across the state. One of our most outstanding employees is Nicole Mode, who was our data manager, and she has a master's. But we call her the queen of data, because if there's anything you need to know about data, if there's any statistical problem that you have, she's the go-to person. She's co-author on many of our papers. Sometimes when we just have an idea, we will say, let's get Nicole in here. Let's get her thinking about how we should structure this. We have PhDs. My staff scientist, Nikki Norenhooten, who runs the lab. She's a PhD. She's really a cancer biologist, but she sort of got interested in what we were doing and joined the team and learned DNA repair, learned health disparities. And so this fluidity of intellect, I think, is probably the most important thing that characterizes the people that work for him. And we have summer students who have contributed. We have post-bac fellows. You know, post-bac fellow is somebody who's finished their undergraduate degree and is taking time off to explore and do research because they're trying to figure out, well, do I want to get an MD? Do I want to get an MD PhD? Do I want to get a PhD? And they spend 2 years with us and they sort of wind up figuring that out. I didn't show the slide because I knew since we had gotten off to kind of a weird start. I have a slide of our post-bacs who are now in MD, PhD, and PhD programs, you know, who are still interested in health disparities, but are going to come at it from many different ways. So, you know, I would encourage all students to get as well trained as you can, but also think about, well, I have a skill set, where do I want to take it?
Well, perhaps we'll close there with that inspiring of all the possibilities that you've shown the students here and the rest of our community. I just really want to say thank you again.
All right, well, thank you for the invitation. And thank you, Bob, for saving the day and running the slides. Teamwork.
Great talk, Michelle. It was wonderful talk. Thank you.
May you live long and keep this work going, Dr. Evans. Thank you.
All right, well, thank you so much. Thank you very much. I enjoyed it.